Morning, everyone. Thanks for joining us. I'm Diane Lipscombe, and I'll be co-hosting this Carney Conversations with my colleague, Chris Moore. This is actually the last day of the Alzheimer's Awareness Month, which is November, and we thought that it would be just a great way to close this month out. Not that any of us will forget the importance of Alzheimer's disease research, but it was a month to really reiterate the incredible need for new cures and treatments and diagnoses in this field. And so Chris and I are very delighted that we have the opportunity to talk with Dr. Alvin Huang and Dr. Hwamee, who are both experts in the area of Alzheimer's disease research, but researching at different levels, and we thought it would just be a really great combination, and we'll get a chance to jump in to hear about their research and their opinions about what it's going to take to find new cures and treatments. And I just wanted to put a little framing on this. I think we all know the incredible need in this field. Every single one of us is impacted in some way or another by Alzheimer's disease. It's incredibly prevalent. 1 in 3 seniors are going to die with Alzheimer's disease or another kind of related dementia. And it's really staggering to think that's going to be more than the number of individuals who die because of breast cancer and prostate cancer combined. I mean, this is a slow, chronic, growing, cumulative disease that really is in desperate need of new approaches to really mitigating the impact of Alzheimer's disease. Even just kind of changing the trajectory just a bit is going to bring relief to millions of people. Right now, 6.2 million people in the United States are living with Alzheimer's disease, extraordinary number, and that number is going to double by 2050 if the research community doesn't bring new diagnoses and cures and treatments. And so that's really going to be the focus of the presentation today. So I'm going to give a quick introduction to our guests, and then we're going to take it away and begin our conversation with them. So Dr. Alvin Huang is the GLF, Translational Assistant Professor of Molecular Biology, Cell Biology, and Biochemistry. And he joined Brown in 2019. He got his MD in neurology residency from the National Taiwan University in Taipei. And he chose to do a subspecialization in neurodegenerative disorders, hence his focus, I think. His PhD, he also did a PhD, and that was at Johns Hopkins University School of Medicine, and he studied gene regulation in the context of memory formation. And then for his postdoc was at Stanford University working with Thomas Sudoff. And that's where he got very much involved and kind of carved his own way in that lab. Really brilliant set of research accomplishments there, innovating in stem cell technologies and uncovering cellular mechanisms important for brain function in health and disease. So obviously his current research is focused on Alzheimer's disease and aging, and he's just been a tremendous, wonderful colleague to have in the Brown community. So welcome, Alvin. Dr. Hwamee is also an assistant professor and she's in the departments of psychiatry and human behavior, as well as cognitive, linguistic, and psychological sciences. And she's been strongly affiliated with the Carney Institute for Brain Science, as well as Alvin. Dr. O received her PhD in biopsychology and with a concentration in human cognitive neuroscience at the State University of New York at Stony Brook. She did her postdoctoral training in the Helen Wills Neuroscience Institute at UC Berkeley. And she studied cognitive, structural, and functional alterations in preclinical older adults with beta amyloid deposition, which is a pathological hallmark of Alzheimer's disease. And she's really just a tremendous scientist and has really become an expert in using non-invasive methodology to peer inside the human brain, to be able to define really critical correlates of Alzheimer's disease, and is a real kind of both Alvin and Hwamee, I think, are real great trailblazers in joining, connecting different disciplines, and we'll talk a little bit more about that. But just wonderful to have her here talking today. She's using, as I've already said, interesting cognitive neural changes due to abnormal aging and Alzheimer's disease pathology. And so we'll get into a lot more detail about the neuroimaging methodologies that she's an expert in, and critical methodology in the diagnosis of Alzheimer's disease. So thank you both for being here. Thank
you for having us.
Wonderful to be joining you again in this great conversation. So I'm just going to kick off with the first question, then Chris is going to jump in afterwards. But I think it would just be wonderful for you both to give us, the audience, an introduction to your research, and perhaps there's a moment that was inspiring to you, like led you to the focus that you both have, and really influenced your current research. So maybe I'll start with you, Alvin, and then move on to Hwamee. So
okay. Well, so for me, my interest in Alzheimer's disease was indeed prompted by my own grandma's early diagnosis of Alzheimer's disease. So back at that time, I was still in my native Taiwan and on my way becoming a physician. So I would say my grandma's last few years, despite, they were very stressful, particularly to my parents, but they were also intriguing and inspiring in the ways that they really prompt me to choose neurology as my specialty. So I still remember, at the time is that, so for example, one thing I vividly recall is that, so my grandma, he would not remember anything we just talked about five minutes ago, but at the same time, she vividly remember all the details happens decades ago. So for example, one thing that's deeply engraved in my mind is that, so that day, she was very eager to share her fond memories about her 3-year-old grandsons. And telling how adorable and how much she loved him, but without realizing that the little boy was standing right in front of him, in front of her. So, fortunate for us is that my family has a strong support system, and then and we have a relatively good ending. But to me, this opens up the door for me to really try to figure out how our brain functions, and particularly how we form our memory, and how come we will lose our memories so easily, particularly when we age. And then, so after a few years of practicing and then really experiencing the very limited treatment powers I have, and then realize how little I can impact the disease cause of this patient population, I decided at the time to take a detour, just try to understand more fundamentally how our brain operates globally. And then because to me, that is the only way they can provide the best chance for us to correct or restore the impaired cognitive function. But at that time, what I didn't realize is that detour now become a direct path to my current research career. So as Diane mentioned, for my PhD, I work at the very basic gene regulation mechanisms in the context of memory formations. But in my postdoc, I decided to take my long-term motivations and then use my basic knowledge and try to understand the molecular mechanisms behind Alzheimer's disease. And in particular, I try to learn the new technique and to develop new model system which is based on stem cells. So in this way, I will be able to study how exactly is happening in human neurons grow in a dish. That's in response to a lot of conditions that we know promote the development of Alzheimer's disease. So my main focus is on the risk genes and or the risk factors that got carried in our own genomes. And because to me personally, it's the most exciting discovery in the past decade for Alzheimer's disease has been driven by our advances in genomic sequencing. So we now have identified literally hundreds of genes that's linked to either increased or decreased odds that can be predisposed to Alzheimer's disease. And then so my research focus is on using the human neurons as well as the human brain cells derived from the human stem cells, try to understanding the functions and then the influences of these newly identified risk genes in the development of Alzheimer's disease.
Great, thank you, Alvin. That was a great journey from your very earliest memories to what you're actually doing in the lab now and the motivation. It was really fabulous to hear.
Yeah, and one thing I like to mention is that, that as Diane said earlier, now is the Alzheimer's Awareness Month. I think this awareness is really, really important because back to that time, we just didn't have this kind of awareness. So my memories has been very retrospective because, now I got all of my medical training. I mean, my grandma's early symptoms are just like the textbook, very classical one. But none of us at that time were able to identify that because we just don't have awareness we needed. So looking back, if one thing, I mean, we can do it better is that if we have this awareness, and then we will have better understanding, and then better sort of handling of our situations, and we certainly would suffer less on that. So I'm so happy that we have this global movement on the Alzheimer's awareness.
Yeah, thanks for underscoring that. I think that's really a really critical point. So Hwamee, go ahead, tell us about your journey.
Yeah, Diane and Chris for inviting me to this event, and it's great to be here with Alvin. Huang, who is one of the leaders in Alzheimer's disease research, the topic that is so important and relevant to everyone. Briefly, my lab currently, as Diane introduced, to try to understand the morphological and molecular and functional changes during brain aging in Alzheimer's disease pathologies and their impact on human cognitive system, particularly memory system in cognitive normal older adults. So it sounds very, when I say about Alzheimer's disease pathologies and cognitive normal older adults may sound a bit contradictory, but this is kind of the population that is called preclinical Alzheimer's disease. So now we know there's a large sort of temporal gap between the development of pathological changes in the brain and the expression of clinical symptoms. So I'm studying this Alzheimer's disease from the human cognitive neuroscience perspective using non-invasive human neuroimaging method. But I was not sort of the human neuroscientist from the beginning. Actually, my undergraduate major was in English literature and education. And I spent for 4 years in that topic. And then I applied to the psychology graduate program I got very, very interested towards the senior years about the neurotransmitter system. And I got very, very excited about the brain that I haven't felt that kind of passion towards the English literature. So I at that time, I didn't know what kind of area that I'm going to be studying in neuroscience. It's a huge area. And then during my master's program, my friend and I got an opportunity to be the part of epidemiological study, which was the first epidemiological study of senile dementia in South Korea, where I originally from. And during that study, I was administering a battery of neuropsychological tests to older adults who living in the community but come to the study site for their participation. And that study, I had a chance to observe a very, very sort of the phenomenal features that I wasn't aware before. So for example, one lady I clearly remember, she was very, very well groomed, about in 70s, and came to the study site for neuropsych testing with her husband. She could walk, she could talk, and she could eat, but she, couldn't, as Alvin talked about his grandmother, couldn't repeat back to me a couple of words that I gave her to report back to me. And also when I showed the bill of $1 bill, something like that in Korean monetary form, she couldn't recognize how much it is. So even though I was new, I was aware that there is a neurotransmitter system, something goes wrong, then those, the human behavior and cognition may change, but it was very shocking to me that how come the motor aspects or other aspects are normal, but selectively they impair in certain cognitive domains. So that triggered me to get to know more about how the brain works and how they are functionally specialized. So I came to the United States to actually study the brain function. But during my graduate study, I, in part because of the funding, my research was more focusing on understanding human memory system in healthy young adults using human neuroimaging method called functional MRI. But during my postdoctoral training, I got into the lab that pioneered postural emission tomography neuroimaging method, another neuroimaging method studying aging brain and Alzheimer's disease. So I had a fantastic and wonderful postdoctoral research experience. And from there, I continued to work on this field.
Oh, wow. Thank you, Hwamee. Amazing. And I'm going to kick it over to Chris, but I just wanted to make one comment, which I think just suddenly struck me as you were both talking. I mean, you're both kind of at the early stage of your independent careers. And it isn't that recent that there have been resources to study Alzheimer's disease. Like that struck me with what both of you said. So there was a lot of resources in what is memory, memory mechanisms. But it hasn't been that long since, there's been this awareness and this important influx of funding and resources for the community to get around this gnarly, really complex, but tractable. Like this, we're going to understand this disease so much better now with the kind of resources that are available. And so, like I think that just came across in what both of you are saying in very recent times. So
right.
Yeah.
So I sort of briefly mentioned about the neuroimaging method that I'm using for my research. I study humans. I wanted to study humans, and I think that it's very important to study humans for Alzheimer's disease, because Alzheimer's disease, there's some kind of debate whether this Alzheimer's disease is very unique to humans. So in some sense it is. Until about, so the Alzheimer's disease is actually clinically diagnosed, characterized by a debilitating progressive impairments in multi-domains of cognition, but pathologically characterized by the presence of two pathological changes in the brain. One is called the abnormal aggregation of beta amyloid proteins, known as senile plaques, and the abnormal aggregation of phosphorylated tau proteins known as neurofibrillary tangles. And those pathological markers, people used to know that those are the information that we can get from autopsy when a patient passed away, and then the brain tissues are stained. So that was sort of considered as gold standard, but we couldn't know until actually the patient passed away. So even though the patient was diagnosed as Alzheimer's disease before they passed away, there is always a chance that they might have been misdiagnosed. And those autopsy, the neuropathological markers could be kind of confirmed much later once the people passed away. However, in 2004, that was the first year, actually, the methods that has been developed as a called the tracer that is used during the positron emission tomography PET scans to actually allow us to image the those pathological changes, the amyloid plaques in the brain. So it was almost Alzheimer's disease. The Aloy Alzheimer's actually reported a case of the first Alzheimer's disease patients about 100 years ago, before this first amyloid tracer has been developed that allow us to image the pathological changes in living individuals. So there was a huge sort of a change that actually allow us to actually track the changes in the brain before they passed away. And another, only about 5 years ago, we didn't have the neuroimaging tool that allow us to actually image the neurofibrillary tangles, which is another pathological change in Alzheimer's disease in living individuals. And those tracers has been developed only about 5 years ago. So now we do have the tools, neuroimaging tools that allow us to image the pathological change in the brain. And now we know or confirm that has been already shown in autopsy studies that these pathological changes actually occurring much earlier and even before the clinical symptoms begin to show. So there is a gap about 5 to 20 years between the development of the amyloid plaques and memory impairment and the loss of independent daily functioning. So there is a 20 years a long age, the time gap. So there is a room that we can intervene during the early stage of the pathological changes.
Yeah, no, good point. Thank you. So Chris.
Well, that begs a question that I think a lot of us are really deeply interested in, or I know that a lot of us are deeply interested in, which is that gap due to the fact that amyloid is an exhaust fume of Alzheimer's? In other words, it obviously predicts the ultimate acquisition of a full-blown disease state, but it's not the engine driving the disability that comes with it, or is it in some subtle way engaged? I mean, maybe you could, so Hwamee, maybe continue your momentum on this.
Right.
So talking about, is that really the driver?
Yes, so if I say it is a driver, then people would think that, oh, I'm kind of a supporter of amyloid cascade hypothesis that is highly sort of on debate currently. But I'm not sure, but even though those pathological markers, such as particularly in amyloid, that are present in the brain, does not necessarily mean that those pathological changes cause the disease. So it can be epiphenomenal. I don't think we know for sure yet. However, in the animal models, by a lot of manipulations, people try to sort of show the link, the causal link between the amyloid proteins up to the memory impairment, and through the synaptic loss and cell loss and tau accumulation. In human studies, I still think that it is open question whether actually amyloid causes some toxic effect in cognition, but it is not strong, that's for sure. And also, However, the amyloid, 100% of Alzheimer's disease patients have amyloid plaques in the brain, but not all tau pathologies cause Alzheimer's disease. So there is a collection of neurodegenerative diseases that is caused by tau pathology, collectively known as tauopathies. Those diseases develop tau pathology without the development of amyloid plaques in the brain. So for example, frontotemporal dementia, which is another type of dementia, does not necessarily have amyloid in the plaques in the brain, but they show the symptoms associated with frontotemporal dementia, mainly due to the accumulation of tau pathology. However, in Alzheimer's disease patients, they do have both amyloid plaques and neurofibrillary tangles when they show the severe clinical symptoms. Thus, there seems to be something going on with amyloid. It's unclear whether it causes directly the memory impairment, but it seems it is still in the pathogenesis process that may play a role in tau accumulation and spread of the tau pathology.
Absolutely. I mean, if you follow the thread of the ultimate driver of the accumulation of these signs, it would strain credulity that it won't eventually lead to insight about the disease mechanism, as many people are exploring. But it's, the recent clinical trials are, and the difference in the response of mice, as you're saying, it's maybe a uniquely human disease, a little bit begs the question of at least the linear interpretation that the amount of amyloid linearly equals the amount of deficit or easily equals it. I don't know. Alvin, did you, in terms of markers for the disease, are there, what is your thought on, this is the reigning view in the field, right? Did you have particular thoughts on,
well,
from your perspective of the elephant, the part of the elephant that you grab?
Yeah. Okay. Well, I think I would definitely second what you and Hwamee talk about. Is that there is a gap. There's clearly a very long window between where the disease starts and where the symptoms sort of appears. And then, so of course, Alzheimer's disease, we know they are hallmarked by the first characterized was the amyloid pathology. Therefore, Hwamee details a lot earlier. And then the linkage between the tau pathology as well as the disease system. So I think that, while these correlations or the associations are very clear. So now we are trying to, at least from a basic scientist part, we are trying to sort of swim against the flow and to try to figure out what's happening in the more like upstream causes. So now we come to realize that there is indeed a very wide range of cell biology process can get involved into the process. And then they will eventually lead to the buildup of amyloid or tau pathology. We can detect that. So so what the question is that, so what would be the sort of earliest driver for this disease, the process that? So I would say, so our recent researchers in the past decades have identified new and important and to me very understudied mechanism to drive this process. So for example, inflammation mechanisms. And then there are also new evidence pointing to some like lipid functions, and as well as some mechanism that's involved into how the protein or cellular contents got traveled and transported inside the cells. We call endocytosis. And then and more ultimately, there are also evidence showing that maybe the drivers for the disease can be just decided by the individual's genomes, and then so I think it's fair to say that now we know more mechanism can particularly precipitate the disease. but we also get more confused about, then which one will be the right one we should target at in terms of to develop a next generation treatment.
Yeah, and also in order to add some about those failed clinical trials in humans when the amyloid was targeted. So those studies were proceed in humans because animal studies findings was so robust and very, very clear. So there's a clear gap between the animal findings and animal models and the human population. And currently, the clinical trials that has been failed in human subjects, most of them were targeting very late stage of Alzheimer's disease patients. Once they diagnosed in dementia, mild dementia or moderate level. However, as I mentioned, these amyloid pathology accumulates very, very early, about 20 years, 15 years before the clinical symptoms, and then reaches asymptote, which means it doesn't accumulate anymore. And then right after that event, other pathological changes, particularly the tau pathology, begin to ramp up. And the tau pathology is critical. Without tau pathology in the brain, with only amyloid, actually it's not going to develop into the clinical symptoms of Alzheimer's disease. So it's important to when these amyloid targeting drugs are sort of tested in humans. And also in light of the current, recently FDA approved drugs that is also amyloid targeting drugs. I think the debate may be not only about the amyloid cascade hypothesis. I think the more debate is depending on how to interpret the clinical the results from the clinical trials. Some people weigh in more the positive results, whereas others may consider those positive results as very non-significant or other factors. So those are more on that debate rather than the amyloid cascade hypothesis, which is, I think, still valuable to be tested.
Thank you both. You in your in this last discussion, you both very clearly mentioned gaps in knowledge, gap needs, and so I think it would just be really great to dig into that. So maybe I'll flip back to Alvin, but as we're thinking, you both were also emphasizing the importance of getting in early, and I think we all intuitively understand the incredible importance of that. Like, what are the early markers of the disorder? The earlier you can intervene, the more likely it is that one can, at least mitigate, slow down. I mean, I was talking to someone in the pharmaceutical industry a few years back, and he pointed out to me, for the most part, this is a disease of the elderly. Not for everybody, but for the most on average. And if you can just change that trajectory, like it's just a bit, like that's going to have a huge impact on millions and millions of people. So, like I wonder if you could perhaps dig in a little bit. Where do you see the gaps? If you had an unlimited number of resources, Alvin, where would you go? Like, what are you going to do? Like, how are you going to, what are the gaps that you're just like really focused on? How are you going to really bridge between what we know on the basic side and the diagnosis and the treatment in humans? That's a small question, I know. I just love to hear about your thoughts about gaps and needs, and yeah.
Well, I think Diane, you just like cut directly into the core of the biggest problems that AD research community has been facing for decades. But if I can, I think I would like to respond in two different ways. So first is that what would be the earliest like disease indicators, and this is a relatively easier question because we can be informed by the recent and ongoing effort, even from our own colleagues at Brown, that so we know that the closest and then the samples they can tell us and inform about the brain conditions is indeed a tap into the CSF, cerebrospinal fluid. And then so people have been trying to search for markers that are present in this CSF, and then to find out if anything they can tell us and even predict the disease cause. So after like a piling up of many independent studies, there definitely are certain markers or certain classes of markers like standing up. So I think right now the consensus is that the earliest possible one that you can detect just based on the CSF samples are a group of inflammatory proteins. So indicating that a dysregulated inflammatory responses may be happening at the earlier stage. And then there are also some types of biomarkers that tell the different part of the story. So for example, a group of biomarkers.
Sorry, Alvin, do you mind if can you just like define what you mean by inflammatory markers?
Yes.
Like, I think that can mean different things to different people. I know what you mean,
yes.
but
yeah,
like,
well, yes, so that's something I'm currently working on. So, obviously I will get very excited when I'm talking about this. So the interesting about the brain's information process, I mean, we call it neural information, is because that it's very distinct from the knowledge we know in the peripheral immune system. There are triggered and mediated by the T cell and B cells that are much better known. So in the brain, the process of this neural information is initiated by the brain innate immune cells. So they are glia cells, astrocytes, and microglia. So all these inflammatory biomarkers that we talked about are secreted or expressed by these cells. So these markers, we know little about their exact function in the brain. But if we tap into the knowledge from the peripheral immunity, we know that they function like a signaling mediator and try to convey important message in the process of our cells in response to stress or damage, and how that would clean up the mess and then to repair. And so this part of the inflammatory biomarkers findings indeed is very supported by the genomic studies that I mentioned earlier. Is that so? One biggest discoveries that was made independently by groups across the globe is that so a group of genes, particularly or tied to increased risk for Alzheimer's disease, are the genes they only almost exclusively expressed in these brain immune cells, particularly in microglia. So it points to that, even though we don't know what's going on with this microglia, but it's very clearly that some process or some responses of these immune cells go wrong, and then initially it probably only makes a teeny differences, but it's really the chronic accumulation of this tiny difference along the time course of up to decades. They will eventually drive a more apparent and more relevant AD pathology as amyloid and tau that Hwamee just mentioned. So I think that the biggest gap here is that, so we know that something going on like decades before symptoms can develop, but how can we identify the true culprit? And then to apply a treatment or prevention strategy at the time that's early enough to make the changes. And it's difficult in a way is that we know inflammation is indeed a very critical part of our body's functions. We need this to maintain a physiological homeostatic conditions that our brains needs to operate properly. So so the problem is how, it's we really need to like fine tuned into the range. They can that's more like a pathogenic or more problematic for the disease, but without interfering its normal function in the brain. So to me, that is the hardest part. Yeah, because when it was first when people are informed by this exciting data showing that okay, information really holds the key, there's a lot of tests and experiments and research going on trying to target that. But what we learn is that if we try to sort of abolish or kind of deplete this function or this type of cells, we actually do more harm than help for this disease process.
Yeah. I mean, is there a way, Hwamee, to, say that take Alvin's, we know Alvin works in this area.
Right.
But
so
like, there's so much information out there to indicate like the earlier signs, at least if you're looking at CFS or the neuroinflammatory markers. So
right. So
is there a way to image that? Like, can you image that? And
yes, actually, I'm very looking forward
to
be able to image the microglia activation using your human neuroimage method, which is actually possible. Just on the tracers is not widely available. Hope that the tracers can be developed that is widely available so that we can do the study in both the humans and in collaboration with Alvin in animal models. But I think there is a general consensus. So how the clinical definition of Alzheimer's disease and pathological definition of Alzheimer's disease. But I think that the people still trying to figure out is what causes the disease. As I mentioned, I think the pathological markers do not necessarily mean that causes Alzheimer's disease. So in terms of the causal factors of Alzheimer's disease, it can be multifactorial based on what we see in human studies. When we look into the risk factors, that may tell us some information about what might be the causal factor of Alzheimer's disease. So hypertension, a lot of systemic diseases that people have in middle age, actually increases the risk of developing Alzheimer's disease in their later life. Diabetes is one of them. And also recent studies also show the sleep dysfunction or sleep sort of dysregulation is also a huge risk factor of developing Alzheimer's disease. So what does sleep do, and how it literally shows the lower level of amyloid accumulation or higher level of amyloid accumulation depending on the sleep that people have. So that was another huge risk factors. Other risk factors includes the sort of lifestyles that also can be modifiable. So those all lead to some ideas what we are still do not have a clear understanding of, and also will be helpful to develop the prevention intervention and strategies if we have a better understanding about those links between risk factors and Alzheimer's disease.
Yeah, I have to say it's incredibly exciting talking to the two of you at this great relatively early but definitely off the ground point in your path, and to think you're both here and because it's so clear there's this multi-dimensional answer to if maybe we could combine certain kinds of minimally disruptive, affordable whole brain imaging methods, and then pair that with blood. Like, it feels like there's going to be a multi-dimensional bioinformatics answer to something like predictive load of risk, and then people might find it worth it to take the risk on certain treatments. I don't know, that's just a comment. So Hwamee, I'm really interested in the sleep connection, as I'm sure as someone who had too much salt and didn't sleep well last night, and you mentioned hypertension, maybe that was TMI, but still, there is a really neat factor in the brain, which is that the brain, the spaces in the brain that allow clearance of waste products of its use. When cells are active, they do create quote unquote garbage, as it were. And there is some really interesting evidence that at night, the actual spacing in the brain changes by up to 60%. It's a huge change relative to how fixed you think your brain networks are. And maybe there's better clearance at night. That's the idea. Is that a reasonable view? Is that something that's getting support? I mean, there's some controversy over the details of that view, but there isn't controversy over the fact that at night there clearly is greater clearance. But I don't know if the link to Alzheimer's is a tight one yet.
Right. I think that, if the amyloid is still considered as part of the pathogenesis and early stage of the markers, regulating or, sort of either reducing the production and secretion of amyloid proteins from the transmembrane of the cells into the extracellular space, or clearing those beta amyloid proteins circulating in the brain might be the two ways actually to deal with and lower the amyloid accumulation. So in terms of the clearance and the role of sleep in the clearance, it has been quite shown. There is a system, as Chris mentioned, about the lymphatic system that actually circulates the amyloid proteins and then moves out of the brain to the body, eventually through the blood stream. So, and that circulation actually goes up during sleep. So that might be one mechanism that scientists have already shown. And another mechanism of the sleep is actually, there is a beautiful study in transgenic mouse models that show the amyloid proteins in the brain actually mimic the diurnal patterns. So during the daytime, it goes up. During sleep, it goes down. It really, really mimics the sleep cycle and diurnal cycle of the mouse. So those are two very, very strong evidence that underlies the biological mechanism of amyloid production and secretion and the clearance that is happening in relation to sleep. So it becomes a very, very important part of our already busy life to maintain very good sleep, actually.
Yes, the very busy life part is one I'm acutely sensitive to when you say that. So is it the case, and this we've had some great discussions leading into our call here. Maybe we'll start with Alvin. But so if Alzheimer was a person, right, and a person who found a really interesting phenomenology, which was this memory loss and then the change in the brain, that's fabulous, and discovery happens that way. But the same way that we're learning that, gosh, there's so many different markers and probably need to be viewed in so many different, throwing large machine learning at the problem and showing lots of logic in the problem. Alvin, maybe start with this, but if you both could comment, is Alzheimer's a disease? Is Alzheimer's 20 diseases? This is begged by the question, right?
Yes. So I think, Chris, I'm very happy that you mentioned one of the biggest challenges in the field is that, so in this disease population of Alzheimer's disease, that we have observed a huge level of heterogeneity out there. So yes, Dr. Alzheimer was the first person to report very classical pathological findings in human brains. So that led to the diagnosis and classification of this group of cognitive diseases. But however, more and more we realize that, so for many of the patients or the brain samples, they shows this very common or like a hallmark pathological changes. They may not share consistent clinical symptoms. Just like Hwamee said earlier, that with this powerful tracer to identify like amyloid load, you can still see like some normally cognitive peoples, they have a buildup of an incredible amount of amyloid, but without showing any single sign of dementia. And then, and one thing, you know, from a basic researchers, I would like to point out is that, so I would say that the 95, almost like a 90 to 95% of the knowledge we know about the molecular and cellular mechanism of Alzheimer's disease are derived from some very rare mutation of the genes, like amyloid precursor proteins, as well as a lot of enzymes that are responsible to process these precursor proteins and then to generate A beta. So that led to the amyloid cascade. But we also realized that, particularly in the very recent age of genomics, so there's also clear evidence showing out that, so people who suffer from this familial or hereditary early onset Alzheimer's disease, in terms of their brains, like gene expressions, are very different from their counterparts. They suffer from the more like a general, more like an idiopathic late onset disease. So, so I think, you know, response to Chris' question, I think a better way to call Alzheimer's disease is to call it Alzheimer's diseases. And then, and to me, this can definitely provide a more nuanced approach, particularly to provide the right sort of prevention strategy, as well as the treatment strategy that will be needed. Because, you know, a lot of times I think that the confusion or the inconsistency in the data they got published probably can be account for this heterogeneity in the populations they got to sample. And then and overall, we know we are marching toward this era we call precision medicines. That means that maybe, you know, in the near future, a treatment of many diseases, including Alzheimer's disease, would have to be personalized in the ways they can target potentially like a very distinctive cause of disease that just, you know, may not be that easily identified by single disease marker or pathology change. Yeah.
Mm-hmm. And also, for me, I think the amyloid markers, knowing whether the person, if about 60-year-old man comes to me and asks what might be the best way or what information I need to know better early than late, might be still, despite all those debates, knowing whether the person has elevated level of amyloid in the brain or not is important, because the studies have shown, although it may not cause it directly, predicts very, very well the highly likelihood of developing Alzheimer's disease later in their life compared to other information that we can gather. And then they have to, once they know they are in that risk, have that risk factor, they may need to monitor whether they have other pathological markers gonna be developed in their brain, such as tau pathology. Even though once the tau pathology spreads out to other brain parts outside of the medial temporal lobe, then that person may highly likely to develop Alzheimer's disease in the presence of amyloid plaques, and then the tau pathology begin to spread to other parts of the brain. However, there is also the risk factor, gene risk factors such as apolipoprotein E, ApoE genes. If someone has the ApoE4 allele, 2 copies of them, then they have about 8 times higher likelihood of developing Alzheimer's disease than non-carriers. So there is also the genetic risk factors that highly link to also the amyloid pathology. However, there is also recently found a gene that is very, very protective. So even though the amyloid plaques present in this particular group, the person do not develop the tau pathology because of the gene called Christchurch mutation gene that found to be very protective and sort of prevent the tau pathology spread to other parts of the brain. So this person, even though they have, a person has the very genetic risk, autosomal genetic factors, didn't develop the mild cognitive impairment until very, very late stage, late age. So there is some other protective factors as well, both in genes as well as in lifestyles. So for the early predictor, it's important to know whether this person has the ApoE gene for carriers status or elevated amyloid level, but they have to monitor whether other pathologies are going to kick in.
Mm-hmm. Yeah, I think that those the balance, right? There's no, I mean, I think with few exceptions, there's no single factor. It's an accumulation of factors, and you can have you know, increase odds or decrease odds of developing Alzheimer's disease within a certain time frame. And I think that you really, it was great that you pointed these, at least perhaps mutations, spontaneous mutations that are in some ways somewhat protective as well, which I think is an incredibly it's interesting and important area of research. You know, we always say this, we've got 4 minutes left, which seems completely unbelievable, but like, you know, time is time. It's a unit, and you know, the discussion has been really fun. So what I'm going to do is to just try and jump through a few quick, get a few quick comments from you both based on some of the questions, and you kind of covered a few of these in more general ways, but you know, what I think is like, there was this comment in the Q&A about Hwamee, I think this is for you, like you've talked about the amyloid plaque buildup timing wise. Could you say something about location? Does location matter? Right? Is it,
yes.
and you know, how does that help make predictions or correlate with disease symptomatology? So that's one quick question there.
Yeah, thank you for asking the question.
Yeah.
So I didn't talk much about that area, but actually that is one of the area that my lab is focusing on currently to better understand what actually amyloid plaques does in relation to cognition, because Alzheimer's disease research has been focusing on actually the brain region, so-called medial temporal lobe and hippocampus, because though that brain structure shrink a lot in Alzheimer's disease patients. So people try to link that amyloid pathology with hippocampal function and human memories function. However, actually, ironically, hippocampus do not have amyloid plaques that much in the early stage. It develops actually outside of the hippocampus. So my current research program trying to focus on trying to understand what those amyloid plaques does in actually the brain functions that other brain parts such as the prefrontal cortex and parietal cortex are heavily involved in. So using some novel methods, and currently we are focusing on more the cognitive functions other than the typical human memory function. So it's still open, but still a lot of questions needs to be answered. But quick answer is people begin to see some subtle changes in executive function in relation to amyloid plaques that highly accumulated in actually frontal cortex and parietal cortex, but not in hippocampus when people are cognitively normal. Whereas tau pathology has a very, very opposite topographical pattern, starts to accumulate in medial temporal lobe, including hippocampus, and then begin to spread out to the association cortices, inferior temporal cortex and parietal and frontal cortex. So, however, even in normal aging and even in 20-year-old, tau pathology is found in the medial temporal lobe, which suggests that it is not Alzheimer's disease specific. So those the spaces where it accumulates is very, very important, and try to link those topographical pattern of this pathology in human cognition that has been a lot studied in psychology and cognitive neuroscience.
Yeah, great. Thank you. And I know this is exactly the area that you're that you're working in. So it's a great question. So, I just want to maybe let Alvin just wrap up his comments too. And Alvin, I thought it would be really I'm just going to ask you like this real simple question, which is based on one of the questions in the Q&A, and then I'll just wrap up and thank you both. I know we're a little over time, but so going back to your your comment about inflammatory markers, and I love the back and forth, and it's exciting to know that you're both talking about this and you're going to work together and get tracers for the microglia. This is really exciting, and I think underscoring what Chris said, it is really exciting to hear you both talk about with passion your research, and so incredibly important. But Alvin, so like 20 years is a question, like 20 years is a long time, as Hwamee was pointing out. It's like these markers that are associated with Alzheimer's disease and the dementias. And so look, what do you, if you were to kind of look into the future, what do you imagine, how do you imagine screening every person at the age of 30? Like what's kind of this like long-term vision of, let's say there is a marker, not likely to be 1, but like 10. Like what do you think we'll be looking at in maybe 10 years, 15 years time? What is the screening going to look like?
Yeah, well, so, that is indeed something going on here at the Brown, is that, our memory and aging program just launched a new, like a registry that we are hoping that we can monitor longitudinally a stable population along the times, and then by assaying their cognitive functions and also getting blood to see to see any correlation between marker and cognitive changes. And then of course we would love to do the genomic sequencing to figure out what would be the genetic risk predisposed to Alzheimer's disease. So so I mean right now it's still at the trial levels, but in the near future, I certainly would love to see this kind of a registry or this type of assay, and can be included in our regular examination and checkup. And then they certainly will be able to offer us the information we needed for that. And then particularly we are now at the era that we are able to handle the huge amount of the clinical data sets that's from your genome, your cognitive, as well as your education, lifestyle, that kind of things. So I believe that all of them can contribute to the disease, but that also means that we would not expect that single item of them would like outweigh the rest of them in the entire population. But in some certain individuals, for sure. But we certainly need to. I mean, personally, I would love to see such data will be available some days for us to dig into it and then really provide some strategies. They can help us, just like a COVID, even so we may not cure them like right away, but if we can flatten the curve, just flatten the curve a little bit, then we will make everything, bring everything to a manageable level. Yeah.
Yep. Well, thank you for that snapshot into the future. I think you're right, and we've learned a lot about what's possible, right, in the last couple of years with really focused effort. So, we are over time, and I just, again, underscore, thank you both. I know Chris feels the same. This was tremendous. And I just feel very optimistic having heard from you both. And I just want to underscore, like, so much is going on here at Brown. Alvin mentioned that, and Hwamee's mentioned that, but like we've got this new Center for Alzheimer's Disease Research, incredibly exciting, the Center for Aging, the clinical groups who have been working a very long time at Brown, treating patients, doing a lot of amazingly important work, Butler and also Rhode Island Hospital and School of Public Health. So like I think the community is just a phenomenal community, and I thank many of you who I know are participating silently in the background, sending in questions, and many of you are just amazing Alzheimer's disease researchers. And so thanking everyone in the community, very much looking forward to the future, and especially having talked to you both. It's inspiring. There's a long way to go, but you know, this is tractable. You can make changes. Thank you both. Thank you, Chris.
Thank you for having me.
Yeah, it was great.
Great job. Thanks a lot.